So this will be lipid lecture 2. And we're going to go through kind of a diagram, and I'll plug in notes for this. This is a very complicated structure diagram showing perfect anatomical positioning of all the organs, or not. I mean, you can see the liver's down here, right around the colon. That's probably not good. But we're going to go through, see there's 5 steps on there. And this actually came out of a medical biochemistry class, out of a textbook for that. And this is the 5 steps of lipid digestion and absorption, if you want to put that in your notes. 5 steps of lipid digestion and absorption. Okay, we're going to say step 1 involves lingual and gastric lipases. Lingual and gastric lipases. Now, as we go through this, you might want to make a note and say, be able to provide the names of the steps and specific details for each step. So I'll ask you to do that. Okay? Now, we're dealing with digestion and absorption of those lipids, triglycerides, cholesterol, including cholesterol esters, I'll explain that in a minute, and phospholipids. That equals almost 100% of dietary lipids. But of those 3, which one's most common? Triacylglycerols, which is the same as triglycerides. That's about 95% of our dietary lipids. Okay? That's about 95% of our dietary lipids. The other 2 categories make up approximately the remaining 5. So we're going to focus on those as we go through these steps. So in step 1, as soon as you consume lipids, lingual lipase is released from glands under the tongue. Basically, they're the saliva or serous glands under the tongue. Releases lingual lipase. Now, when we talk about these lipases, they basically do this. They hydrolyze triglycerides into a monoacylglycerol, diacylglycerol, and fatty acids. Okay? Lingual lipase tends to do this. Tends to make diacylglycerol and fatty acids. Right? So we know what we're talking about here, right? Here's our triglyceride. Glycerol backbone, 3 fatty acids. So the diacylglycerol is going to tend to have this. That one tends to cleave 1 and 3, I believe. So you're going to have 2, 1, 3. This one tends to come off. This is the diacylglycerol. The monoacylglycerol tends to end up like this. Just the fatty acid on the middle carbon. Now, lingual lipase is interesting. Lingual lipase tends to prefer hydrolyzing short and medium chain fatty acids. Lingual lipase tends to prefer hydrolyzing short and medium chain fatty acids. And those fatty acids are really common in milk. So a major function of lingual lipase is to allow newborns the ability to hydrolyze milk fat. Okay? That's why it has that preference. Gastric lipase is secreted by cells within the stomach. So when the fat comes into the stomach, gastric lipase hydrolyzes some of the triglycerides. Now, neither lingual or gastric lipase has a huge effect on triglycerides. So they hydrolyze some of the fatty acids, but that process is relatively fast when it goes through the mouth into the stomach. And then in the stomach, it's a very acidic pH. And that slows those enzymes down. So lingual and gastric lipases kind of start and perform some triglyceride digestion, but nowhere near to completion. Okay. Got all this. So step 2. And step 3 are kind of combined, or they occur at the same time. And that is when, so for example, in step 2, that's when the pancreatic enzyme release. And step 3, we're going to say liver. What else are we missing on there? Liver releases bile. Gallbladder. Good. Liver and gallbladder. Release bile. We're going to say bile and bile acids, right? So the mixed micelles can be formed, so we'll talk about that. So that's basically at the same time. Now, the main enzyme for triglyceride digestion comes from the pancreas. Okay? But remember, the pancreas is typically not at full capacity until a few months into life. It's usually like 5 or 6 months into a child's life is when the pancreas begins really being able to meet demands. So those first few months, that's why lingual lipase is so important. Lingual lipase is at a high level before birth. As soon as that infant is born, that lingual lipase is ready to help with milk fat digestion. Okay, so let's look at these because I think we've covered enough to go into this table. So this is a really basic table. It says multiple lipases assure efficient intraluminal hydrolysis of dietary lipids. When we talked about the digestive system, I said the pancreas is the most important tissue or organ for lipid digestion. Nothing happens on the surface of the intestinal cells. It's basically coming from the pancreas, and we have some coming from the stomach and some coming from the mouth as far as the lipase activity. So here are the major enzymes. The acid lipases are called lingual. This is lingual and gastric lipase. All right, they have to be able to function in the acidic environment of the stomach. They do a little bit, but not to a really significant degree, but they're referred to as acid lipases because that's their environment. Come from the lingual glands and the gastric mucosa. They function in the stomach and a little bit in the small intestine. But here's the main enzyme for triglyceride digestion. That's pancreatic lipase / colipase. There's 2 enzymes that are required. I'll give you details on that coming up. Comes from the pancreas, functions in the small intestine. We have 2 other enzymes we're going to look at. That's cholesterol esterase and phospholipase A2. They both come in from the pancreas into the lumen of the small intestine where they function. So as part of these steps, you're going to say each step, and you will include the enzymes and products involved in each step. So for example, when we have steps 2 and 3, you're going to say pancreatic lipase colipase hydrolyzes triglycerides into 2 fatty acids and monoacylglycerol. So the fatty acid on the central carbon tends to remain on that glycerol, so you end up with a monoacylglycerol and 2 fatty acids. Now cholesterol ester, I'll show you that. Now, so you might want to put this in your notes. Approximately 15% of all dietary cholesterol is a cholesterol ester. And a cholesterol ester is cholesterol with a fatty acid attached right here. So a cholesterol ester is cholesterol plus a fatty acid. And that fatty acid is attached right here. Okay, it's not on this one, obviously. Cholesterol esterase removes that fatty acid. So what kind of bond is linking that fatty acid to cholesterol? It's an ester bond. It's a covalent bond. Okay, so it removes the fatty acid to help absorption. Okay. The other enzyme on that table is phospholipase A2. And as its name implies, it cleaves a fatty acid from phosphatidylcholine and other phospholipids. You probably talked about this in other classes, but remember, phosphatidylcholine is also known as lecithin. So it's very common that lecithin is in the diet. It can come from animal products as part of their cell membranes or processed foods. If they have emulsifying agents, for example, and it contains lecithin, then that enzyme removes this central fatty acid. And we'll go through that step in a few minutes. And again, why do we do this? Digestion. Helps in digestion. Because digestion in some of these is required for absorption. So step 4 is what's coming next. So now remember, when these enzymes cleave their substrate, so for example, here's the one we just mentioned, phospholipids, the fatty acids removed. When you remove a fatty acid from those phospholipids, it's now called a lysophospholipid. A more specific name would be, so if this phospholipid was phosphatidylcholine, then it would be called what? Lysophosphatidylcholine, because that fatty acid has been removed. And I'll show you a picture of that in a second. Okay? Now these are the hydrolysis products. So this is what ends up. Fatty acids, monoacylglycerol, cholesterol, fatty acid, and then lysophospholipids. Those are incorporated into mixed micelles. We talked about that a while ago. So now you have those mixed micelles that can interact with the surface of the enterocytes. And those lipids that have been hydrolyzed can diffuse into the enterocytes. Okay? So let me show you this. And then I'll go back to that previous slide. So this is kind of where we are at this point. It's probably better if this says mixed micelles. So these are micellar particles. Love it when it does it. So these are meant to show mixed micelles that are bound to the surface of the enterocyte and their contents are diffusing in. So this is some of the things that we mentioned. We said cholesterol can be part of these. free fatty acids, monoacylglycerides, and here are lysophosphatidylcholine molecules and other lysophospholipids. So now those digestion products can be absorbed into the cell. So when you consume lipids, they go into our gut, they're digested, they become part of mixed micelles, they go into the cells, and then they're recombined. Okay? The main dietary lipid is triglycerides. So one of the things that's interesting about this process is we have pancreatic lipase, which hydrolyzes triglycerides into monoacylglycerol and fatty acids. So why do we need this one? Have you heard that term before, coenzymes? Might have heard it as vitamins or something like that. Well, this is a coenzyme for pancreatic lipase. It's called colipase. And this is what it does. The next slide, kind of 2 slides from here. This shows the function of colipase. And this is a slide that I got this from American Gastroenterology Association, so this is not in your textbook. But I think this is a good graphic to show what's happening here. Now, if, for example, you have a fat droplet, and imagine this is in the small intestine. You have a fat droplet, and lipase, so this would be pancreatic lipase, binds to the surface of the fat droplet. It'll hydrolyze triglycerides into monoacylglycerol and free fatty acids. So it'll break that triglyceride apart. So if you want to measure the rate of activity of that enzyme, you just look for fatty acids being released. So if I set this up in vitro, so if I take a test tube, and let's say I put water in there, and then I take triglyceride and I add it, and I mix that up, and then I let that sit for a minute, that fat will basically coalesce once again, and it'll form fat droplets. Now if I add pancreatic lipase, the lipase can bind to the surface of the droplet and start breaking down triglycerides and releasing the products away from the fat droplet. So that's what's happening here. Okay? Now that's not what happens in the gut. In the gut, at the same time that lipase goes into the small intestine, so does bile. And bile includes bile acids. Remember what bile acids do? They emulsify those droplets. So those bile acids bind to the surface of the triglyceride droplets and make them into smaller lipid particles. So that's what these are supposed to be. These are bile acids. And so this has a little charged portion here, and this part of the molecule are the hydrophobic rings. And so this has, for example, bile acids kind of sticking together through their hydrophobic rings until they encounter the surface of the lipid droplet. But look what they did to lipase. They push it off. So bile acids cause lipase to dissociate. Bile acids can push lipase off the lipid droplets. So again, I go back to my test tube, and let's say a few minutes into my reaction, I take sodium deoxycholate, a bile acid. I add that, and I mix it up. My reaction stops. So the release of fatty acids goes flat. Now, if I want to mimic in vivo conditions, I would then take colipase and add it to the reaction. And this is what colipase does. Colipase binds the lipid droplets and holds lipase on their surface. So it overcomes the bile acid inhibition of pancreatic lipase. So look, so colipase binds the lipid droplet and pancreatic lipase to overcome the bile acid inhibition of lipase. So without colipase, lipase really doesn't function. So you have to have that. And that's what it does. I don't know why the system was made that way, but it is. And so lipase depends on colipase, and that's what it does. As soon as you have that there, fatty acid release continues.
Why doesn't colipase and lipase just go in together like in the beginning?
I don't know the sequence. I haven't seen anything on that. Good question. But so colipase has a binding site for the lipid droplet. It's got a binding site for the enzyme. I don't know if it can bind to the enzyme without first binding to the lipid droplet. I know that there's been, in some scenarios, people have genetic alterations in colipase, and that really causes a problem for their lipid digestion. So I don't know if there has to be a sequence or if it's just kind of a random interaction. Got all that? Uh-huh.
So would this be occurring in step 3?
Yes. Yep. Ready? Step 4. This is absorption. Digestion's been done. Mixed micelles are formed. Now the mixed micelle contents are diffusing into the enterocytes. And then it looks like a big mess here, but let's simplify this. Okay? So let's take each part of this, kind of one step at a time, and kind of big picture, what we have to do is those lipid molecules have to be prepared to go into the blood. So they're eventually going to end up in chylomicrons. Okay? But now look at the contents of chylomicrons are triglycerides. This is phosphatidylcholine, so if you want on your notes, if you want to put PC is phosphatidylcholine. For people outside of nutrition, it's politically correct, right? If you're in nutrition, PC is phosphatidylcholine. And cholesterol can go in there, and cholesterol ester can be in that chylomicron. So in other words, if you look at this, these digestion products don't go into chylomicrons. They have to be reassembled. In order to reassemble these molecules, the fatty acids have to be activated. Okay, so we're on step 4, which is absorption. And this is what happens. If you look at the central part of this, these fatty acids are being combined with coenzyme A. And coenzyme A is used by lots of different enzymes. So the fatty acid is now activated. It's activated by the addition of coenzyme A. In other words, now other enzymes can use these fatty acids. Now the enzyme that does this reaction is called acyl-CoA synthetase. It's the one that combines that fatty acid with coenzyme A. Okay? Now, this enzyme only uses I'm going to say fatty acids that are greater than or equal to 12 carbons. In other words, it does not work with short chain fatty acids. So that's why these are an exception. So if we consume foods that have short chain fatty acids, they go straight through the intestinal cell into the blood without being part of a chylomicron. And the reason they do that is because they are not activated by that enzyme. So acyl-CoA synthetase needs long fatty acids in order to recognize them. And that's okay with these guys because they're less hydrophobic than long fatty acids. So that they can go into the blood, they'll bind albumin when they go into the portal vein. So short chain free fatty acids travel through the blood like that. Then they can be absorbed by tissues. They don't need chylomicrons. Everybody else in the chylomicron. Okay? Once you have that activated fatty acid, you can take those fatty acids and add them back to the molecules that they came from. So in other words, you can add fatty acids to monoacylglycerol, get a triglyceride, and it goes into chylomicron. So you can just start here and kind of build backwards. So if this is a monoacylglycerol, this goes right like that. If we have lysophosphatidylcholine, remember we're missing a fatty acid, it can be added back to form phosphatidylcholine. If it was cholesterol, fatty acid can be attached. I'll put this one up here. To form cholesterol ester. So here's cholesterol with a fatty acid attached. And then all of these can go into a chylomicron. That's basically all this chart is showing. Don't worry about this part. You can cross off glycerol and glucose. Now it's in the chylomicron. Now that chylomicron can be released from the intestinal cell into the lacteal. Remember that? So now it goes lymphatic system, ascends the central abdomen, left subclavian vein into the blood. So that's a good view there. Now this isn't perfect, but we'll go with this. They're saying this packaging occurs in the ER. Okay? And then that's how it's secreted from the cell. So now what's happened is that chylomicron has been formed in the enterocyte through a combination of dietary lipids and protein called apoprotein or apolipoprotein. So we're going to talk more in detail about this too. Now we have a molecule that's a combination of protein and lipids going into the lymph and into the blood. So that last part is chylomicron formation. So absorption is going into the cell. Chylomicron formation has to do with secretion. So step 5 is chylomicron formation. And this explains it. So resynthesized triglycerides, cholesterol esters, phospholipids, even fat-soluble vitamins like vitamin D, vitamin E, K, and all those make it into those chylomicrons as well. They combine in the ER, and then they're combined with apolipoproteins. They go from the ER to the Golgi, and then they're secreted. They follow that secretory pathway. So they're exocytosed. Go into the lymphatic system, then the blood. And chylomicrons are one member of a family of lipid transport molecules known as lipoproteins. So other members of that family include HDL and LDL. So chylomicrons are part of that. And that'll be our next step when we go through that. Good there? Okay. And I showed you these before, right? So you've seen those. Okay, so that's the end of the 5 steps of lipid digestion and absorption.
Can you just briefly go over what the difference between a mixed micelle, a micelle, and a chylomicron are?
Sure.
So I always get those kind of all.
Sure. A micelle are basically a homogeneous mixture of bile. So some of these, let's say these are some phospholipids and some of them are bile acids, but it's all bile. So this is just a micelle. You go from that to a mixed micelle. You've added hydrolysis products. So now, for example, you might have cholesterol ester in here. You might have triglycerides in here. Oops, sorry, I messed that up. Don't write that. Now it's a combination of bile and hydrolysis products. And then a chylomicron, so these would be in, for example, small intestine. Chylomicrons, I'll show you the structure in a minute, but chylomicrons are in the lymph and the blood. And they have a combination of, they're a lipid core and a phospholipid shell. I'll show you a drawing of that in a second, yeah. So an easy way to just remember is micelles and mixed micelles are in the lumen of the gut. Chylomicrons are in the lymph and the blood. Okay? Now, we're going to go from, we've gone from the kind of the lumen of the gut, we've gone through the intestinal cells. Now we have to be able to take lipids and transport them through the blood throughout the body. So the problem is those lipids are hydrophobic. So you don't put them right into the blood. You package them into transport molecules. Okay? So lipoproteins are spherical molecules, or sometimes you refer to them as particles. They have a lipid core, which are most commonly triglycerides and cholesterol ester, and they're surrounded by phospholipids and apolipoproteins. So hydrophobic core, hydrophilic exterior, so they can go around in the blood. Now there are 5 classes of lipoproteins, major classes. There's more subclasses. One of those classes is chylomicrons. The other 4 are VLDL, IDL, LDL, and HDL. Very straightforward, right? We should have sued the people that came up with this nomenclature. And the only reason it makes any sense is because that's how you separate the particles. You separate by ultracentrifugation. So they have different densities. So in a research lab, for example, when we would separate these different fractions, it would take us about 4 days to separate them. We'd take a blood sample, we'd separate it into plasma, we would take that plasma, we would put it on a tube, put it into a centrifugation tube, and we would set up a density gradient. And we would do what's called stepwise centrifugation or separation. And the first one that you do is, you take this, let's say I'm working on a rat experiment where I'm looking at something that changes, I think, lipoprotein profiles, like different trace minerals or something in the diet. I take their plasma, right, which is the portion of their blood that does not contain red blood cells. I put it in this tube, then I add something to change my density of this plasma. I increase the density of the plasma so that any molecules that aren't dense at all float. This typically, if you're a lipoprotein nerd, you call this floating up your chylos. Okay? Those chylos end up right on top, and you can see them. So I can actually take that tube, I can take a pipette, and I can just aspirate off the top and pull my chylomicrons, and it actually looks like a layer of fat. So when you do this, your animals did not fast, they had a meal, and you pull this right off. These are the chylomicrons. They're not dense at all, they're mostly fat. Easy to float them up. Then you can just work stepwise. Then you take your chylos off, you change your gradient, you make your plasma more dense, and you float up your next layer. This is called VLDL. This is very low density. In other words, not dense at all. So typically, that means the next layer you float up are your VLDL. You pull that off, spin again. Change your gradient, spin again. What comes off next? IDL, except there's so little IDL, you generally do LDL next. Your last spin, you spin down for your HDL. And HDL is high density lipoproteins. What's changing that density, the ratio of what to what? Protein to fat. Yep. Your HDL are the most dense. So that's why they're called that. And there's other ways to separate them, but that's basically the way you do it in most research scenarios so that you can study each fraction. So each of these different classes of lipoproteins has different functions and characteristics. They each do something that we have to have. So overall, the lipoprotein provide a means of transport, dietary, which is exogenous. Right? Exogenous means from outside the body. And endogenous lipids. So endogenous lipids are those made within the body. And most of the time that's in the liver. So these molecules move around fats in our blood from the diet and fats that are made in our body. And they take them through the blood. And so we're going to look at more characteristics of these. Have you ever seen this? Anything like this? This comes from 210, like a basic intro nutrition text. So you may or may not have seen this. This is, on the left side, it kind of shows the size differences of the lipoproteins. So this is, one way of saying this is, let's say this is the solar system of lipoproteins, and so you looked at the different sizes. See that big purple shape? That's a chylomicron. Chylomicrons are huge relative to the other ones. Matter of fact, if you give a blood sample and I obtained your plasma, and if I held that plasma up and it looked like a very clear yellow color, I know you fasted. If I hold that up and it looks kind of a milky, kind of weird texture to it, almost like fat in there, you did not fast, and those are chylomicrons. They're so big, they give that kind of fatty look to it. So they're bigger than any of the other ones by far. Chylomicrons, if you look what's inside them and what they're made out of, they are mostly triglycerides. And where did that triglyceride come from? Yeah, wherever you ate it. Olive Garden, wherever you went, that's what's in that chylomicron. It's almost all triglyceride. Okay? There's some phospholipid. That phospholipid basically came from enterocytes, and that's what's on the outside of it. And then there's some cholesterol, that's dietary cholesterol, and a little bit of protein. That's not dietary protein. Those are proteins that are added to chylomicrons so they can function. So here's what they look like. That structure up there is a generic diagram that shows what all these lipoproteins look like. Phospholipids on the outside. Why? Yeah, you've got to have hydrophilic and hydrophobic. Remember, phospholipids are amphipathic. So they're fine in the blood, and then they can hide hydrophobic molecules. So you see the surface, we've got phospholipids and a little bit of protein. Again, that's not dietary protein. Those are apolipoproteins. I'll describe those in a minute. Okay? We have triglyceride on the inside. We have some cholesterol that's basically here. And this doesn't show it, but there's cholesterol esters on the inside. So they all look like this, but with different sizes and ratios of protein to lipids. That's what changes their density and their metabolism. Okay? VLDL, the next least dense and largest of these, also mostly triglycerides. So you want to remember that chylomicrons and VLDL are triglyceride rich. Chylomicrons, VLDL are triglyceride rich. Okay? You can see why they're a little more dense than chylomicrons. They got a little bit more protein. And now let's look at these. These are the more famous ones, LDL and HDL. What do we say about LDL, good or bad? You say that's the bad one. HDL is the good one. Look at their composition. They tend to have more cholesterol than triglyceride. So remember, LDL and HDL are cholesterol rich. They are cholesterol rich. Okay? Now, and you can see, for example, HDL, very small, quite a bit of protein, so they're relatively dense. But look, LDL and HDL both have a lot of cholesterol. Cholesterol is cholesterol. The reason one is good and one is bad is one is moving cholesterol to tissues, the other one is taking cholesterol from tissues for excretion. So HDL is considered to be the good one, and I'll tell you why. Because HDL are the ones that are basically helping remove cholesterol from the body. This class has higher HDL cholesterol than the general population. I can tell you that right now. I can tell just by looking at you. Why can I tell that? What's that? Because you're nutrition majors and mostly women. Women tend to have higher HDL cholesterol. That's one of the factors that probably leads to you guys living longer. Right? Tell I'm kind of a little bitter about that. But that's why. HDL, and you guys will measure it next year in 329 lab, and we can actually see that.
Why do women tend to have higher?
There's a few reasons. One of the reasons is estrogen. Estrogen tends to increase HDL production because women pre-menopause tend to have high HDL levels, and then post-menopause things kind of change as far as lipid metabolism in women. Guys, we tend to suffer longer, just chronically, slowly die because we don't have those big changes in lipoproteins like that. Yeah. Okay, so we're going to talk about why all that kind of works like that. And we'll look at this structure in a second, but I think this is probably a good time to take a break. So let's take 10 minutes and then we'll continue.